Welcome to The Factor, a podcast series for the life sciences industry powered by Kymanox. Today's episode is hosted by Ryan Doxey, Senior Director of Technical Services at Kymanox, who's joined by David Harrell, President and CEO of Helixomer. Helixomer is a drug design and development company that specializes in RNA origami. So Ryan and David will be discussing the innovative potential of RNA origami in transforming drug development and precision medicine, and also the challenges faced by startups in this complex landscape. To start, let's answer the question: what exactly is RNA origami? Here's David.
Everybody's familiar with paper origami, clearly.
Yeah.
But origami has had a huge impact in all sorts of arenas, from the automobile industry with airbags. They're folded up in origami, to even space, right, where you have self-assembling satellites that are origami.
That's really cool.
So NASA astronauts aren't assembling satellites anymore. They're being launched out and they open up in space. And Helixomer is bringing that concept to the design of drugs.
Oh, wow. Wow. That's amazing. And so, the initial indications for this RNA origami, can you elaborate on that a little bit?
Absolutely. Our initial focus, our proof of concept drug, is a drug pairing. It's a profound, reversible, linear dose response anticoagulant that's non-immunogenic. And that's part of the beauty of RNA origami. We use a modified RNA, which does not stimulate an immune response.
Okay. That's really cool. That's really cool. And so you guys are in the preclinical stage. You're a spin out from NC State.
We are,
right? So the technology was developed at NC State, and now you're getting into the preclinical space in hopes of filing an IND here soon. So let's talk about some of the challenges with this novel, RNA origami, and bringing this into the preclinical space in anticipation of the regulatory expectations. And so what types of studies are you doing now? What are you looking at in terms of generating this body of data to demonstrate the efficacy of RNA origami and how it could translate into humans?
Sure. If I back up a moment and think about our drug is not just RNA origami.
Okay.
Right? We functionalize our RNA origami with aptamers. Right? So both of those are single-stranded nucleic acid technologies.
Okay.
The company was founded in 2020, but you're absolutely right. It was an NC State spinoff. Started in Professor Levine's laboratory at NC State. He had been a molecular engineer, and his early work started with proteins. And then he was starting to think about RNA, but RNA, most people had thought was always going to be unstable. And so people moved into DNA work. Turns out we can actually make RNA very stable.
Oh, wow.
So we modify it with a 2' fluoropyrimidine. And with that, we get not only shelf stability, but we get nuclease resistance. So it's very stable in the body. Now, in terms of the data that we're collecting, we've been collecting data for about 4 years. And again, it's laboratory, some in vitro, some in vivo. We have good data demonstrating not only the efficacy of the drug, demonstrating a linear dose response, but we also have good data demonstrating that it's not immunogenic. Aptamers are pretty small. And so generally with an aptamer, you have to put a bulking agent onto that. Historically, that's been polyethylene glycol, PEG. Right? And there are two aptamer drugs on the market right now. Both of them are for the eye because the eye is sequestered from the immune system. Right? PEG can be really problematic inside of a human body. The eye makes it tolerable. With Helixomer's approach, we've gotten rid of polyethylene glycol. Right? So we now use RNA origami. So one of the beauties of using RNA origami is not just the fact that it's non-immunogenic, the modified RNA origami, but that we can control the spacing between the aptamers and the orientation of the aptamers.
Okay.
So one of Helixomer's mottos, I would say, is that we rescue the promise of aptamer technology.
Okay.
So aptamers were discovered in 1990. The first drug on the market was 2004. But many aptamers that have gone through clinical trials have had problems because of PEG, or they weren't efficacious enough. By increasing and optimizing the spacing and orientation, we drive the efficacy up. And so we actually end up with synergistic effects.
Oh, wow.
So we greatly increase the aptamer's efficacy.
Okay. So the aptamer is the functional.
That is the functional piece.
The RNA origami is the bulking agent that you mentioned.
Correct.
Okay. And so by adding this RNA origami, you're really able to eliminate all of the deleterious effects of the PEG in vivo.
Absolutely.
Yeah, that's amazing.
Yeah.
That's amazing.
And so it's not just a bulking agent, right? PEG is just a bulking agent, but RNA origami, yes, it increases the size for sure, but it does more than just increase size, right? Because we can control all those foldings, right? So that we maximize and get the orientation correct, so that we can boost the efficacy of the aptamers.
Okay, that's amazing. That's really, I mean, that's true innovation, at its best, and the application of not just science but engineering, and it's a great collaboration between the two concepts. So that's amazing. So in thinking about. We have the innovation, we have the product design, we have its intent, and so now we're moving it into the regulated environment, and really trying to demonstrate this proof of concept. So I think you've done a really good job of demonstrating that in vitro with your in vitro models and in your in vivo animal models, and this doesn't sound like it's really from the clinical landscape. Do you think going straight into the clinic, phase one, how do you see this playing into human dosing, and what are your thoughts on how to get this into humans?
Sure, it's a great question. One of the beauties, again, of really focusing on spacing and optimizing orientation is that we can drive dosage lower, so it's safer for patients, number one. We are obviously moving from small animal models to large animal models, which will help us figure out some of the dosing that we'll at least begin phase one with, right? There's a lot we don't know yet.
That's right.
That's part of why we have Kymanox helping us.
I understand. That's great. And and based on my interactions and my understanding of what you guys are doing, I think you're setting yourself up for success and thinking about those risks and challenges, in a proactive manner rather than a hope and a prayer that we get it right. So
I'd also say we have a unique advantage compared to other types of RNA technologies in the fact that we are using RNA for a structural purpose.
Right.
We do not encode anything into the genome.
See, that's a big distinction there.
That's a huge distinction.
Yes. And so, it does give you a different perspective on, the use of the RNA versus, the COVID vaccine and things like that. Yeah.
Yes.
So that's a great point. On your day-to-day, what are the things that keep you up at night? In terms of the challenges that you may face, and where the Kymanox to Helixomer partnership is really starting to blossom.
Sure, absolutely. So, being a startup, one of the things that keeps me awake at night is fundraising. Yeah. That's very different than the regulatory side, obviously.
Yeah.
But I think as we continue to progress in the regulatory space. And working with Kymanox, it makes it less challenging to raise funds,
right?
Because we eliminate certain risks or certain unknowns.
Right.
We evaluated a number of CROs and regulatory experts, and Helixomer really values our corporate values. And I know that might sound like business speak,
Right.
but what we mean by that is our corporate values, we make decisions according to those values, right? So we make hiring decisions or promotion decisions or any other sorts of decisions run through our corporate values.
Yeah, that's great.
And with Kymanox, one of the beauties was that we really felt Kymanox matched and paired well with our corporate values of being smart, of being humble, of being hardworking, and being collaborative.
That's great. That's great.
So we've had a really enjoyable time working with Kymanox.
Yeah.
The collaboration side has really blossomed as we've gone along, and I think oftentimes CROs aren't as much collaborative as Kymanox has been.
That's great to hear. Yeah, that's awesome. Well, I appreciate that sentiment, and we feel the same way. And we're really excited about all of the opportunity to come with this technology, and our mission statement is because our why is because patients deserve better, right? And I feel like that really echoes Helixomer and what you guys are trying to do, to meet the patient need out there. So getting a little bit more technical, and on the CMO landscape, so the contract manufacturing landscape, this doesn't sound like a very easy technology to where you can just drop it in any CMO and expect them to run. So can we talk a little bit about the CMO selection and where you guys see yourself fitting, and what type of kind of bespoke services that you may need?
Well, I would actually argue that it's much simpler than you think it is.
Okay, good.
The same equipment that is used to manufacture the mRNA is used to manufacture RNA origami.
Okay.
So scalability is already worked out for us.
Yeah.
People are familiar with mRNA vaccines. CMOs know how to produce mRNA already.
Yeah.
So it's less challenging than you might think it is, even though this has never been done before.
Right. So that's awesome. And that's a great point. And so, once you get into these regulatory and, controlled environments, and, you have to make the CMO just as comfortable with your technology and instill the confidence in them that this isn't really different from what you've already been doing. So that's great, right? Because you look at the terminology, you look at what you guys are trying to do, and it could be companies could be a little standoffish in wanting to commit to being able to deliver with your technology. But it sounds like it's using what's already out there and then just building upon it to be applicable to your technology.
That's right.
Yeah. That's great.
Unlike mRNA, like the vaccines, which do go into the genome, obviously, we don't need to use lipid nanoparticles. We don't have to wrap anything up. We have a modified RNA that's produced enzymatically, just like the mRNA, which is different than many other kinds of RNA, which are produced chemically.
Right.
Ours are enzymatic. So we're making RNA the same way cells make RNA.
Yeah.
And the fact that mRNA vaccines are so prevalent because of COVID, I think most CMOs are set up to produce
for success. Yeah,
the exact same thing.
That's great. That's great. So moving into once we get this kind of established within the CMO and begin to scale up, and the regulatory bodies are becoming comfortable and confident in your technology. What challenges do you see and think about as you try to progress this through clinical development?
Sure.
Packaging, storage conditions. Is this able to be stored at room temperature or refrigerated? Is refrigeration required?
No. So one of the beauties of using the 2' fluoro modified RNA origami is that we're thermal stable.
That's amazing.
So we can have it at room temperature.
Yeah.
Again, that's very different than other anticoagulants, especially on the profound side.
Yes.
Right?
Yeah. And so, again, just going back, taking the engineering and the science approach to this technology, it seems like, there was a lot of thought that went into, this technology and the end use ultimately. And then not just thinking about, oh, hey, we got an anticoagulant that uses this new technology. You have the lack of immune response, but it's also shelf life stable at room temperature. And so, just building upon that, it seems like you really are, always forward thinking.
We are.
Yeah.
Part of the beauty of aptamers is that they have a very, very precise mechanism of action.
Yeah.
And so Helixomer likes to think of ourselves as redefining precision medicine.
Yeah.
Many times people look at precision medicine in terms of being individual medicine.
That's right.
And we look at it in terms of how the drug works.
Yeah.
So off-target effects are a major problem in small molecules.
Yep.
We have a precise mechanism of action with aptamers.
That's amazing.
So it minimizes off-target effects.
Yeah.
The other part of the RNA origami that makes it really interesting when working, for example, with the anticoagulant, because it needs to be reversible.
Right.
We're talking about profound anticoagulation. The RNA origami is dynamic.
Mm-hmm.
It's flexible. And so our reversal agent actually changes the shape of our anticoagulant molecule to make it inert. And as a direct thrombin inhibitor, it simply releases thrombin.
Wow.
And so you get reversibility almost instantaneously.
Yeah, that is amazing to me when you think about the technology and the applications there. And I'm glad you used the word precision medicine, because I was thinking personalized medicine, but you took it a step further, right? And so you have personalized medicine with the precision that can be applied across the population. So it's
That's right.
personalized, but to the masses.
That's right.
Yeah.
Yeah, which is an interesting, different way of looking at precision medicine.
Yeah, it is.
I think, we've, as a society or a global issue, we've really sort of tolerated the side effects of medications because they're not as precise as we would like them to be, but we need them.
Yeah.
Right.
Yeah.
We've really flipped that on its head.
Yeah.
And, I think National Geographic in 2023 published a journal that said that the title of that edition of the journal was "The Future is Folded," and Helixomer is out on the forefront of that.
That's amazing. That's really cool. Yeah. So let's talk about the drugability aspect of this and where it can be applied just beyond your current target. So can you touch on that a little bit?
Absolutely. So we are looking at a number of arenas, from neurodegenerative diseases, because there are aptamers that will allow a drug to cross the blood-brain barrier.
Yeah.
So we already know those aptamers.
Yeah.
Right. So we can do targeted drug delivery. This can be done in oncology, for example. We can also look at complex protein-protein interactions of what would be considered historically an undruggable condition.
Wow.
And we can change that with aptamers and RNA origami. Now, the other beauty is that RNA origami, because it's flexible, we haven't done it to date. Nobody has, but theoretically, it is quite possible to have a drug take a shape only in a certain environment, whether it's pH or temperature or whatever it might be, and then it becomes active.
Oh, wow.
So there's a tremendous amount of opportunity with RNA origami that we really haven't maximized yet, but we're getting there. And I'd say from a drug design and development perspective, it allows Helixomer to do modular drug design and development, because all we're doing is replacing the pieces of the molecule, right? So we're replacing the aptamer. So I always like to give an analogy that our molecule is like your hand, right? Our RNA origami is like the palm, and aptamers are these binding agents, and they're like fingers. So if we want to hold a big basketball, we'll hold our fingers in a certain way. But if we want to hold a pencil, we'll move our fingers. Yeah. We don't move our palms. We move our fingers.
That's amazing. Yeah.
And so it allows us to do modular drug design, which really speeds things up.
Yeah.
We can carry over certain safety data for sure on the platform that we have. It also allows us to much more quickly go into other disease conditions and indications.
Yeah.
So we're looking at oncology. We're looking at antiviral, antimicrobials.
Yeah.
There's some work being done right now with aptamers that are being used for diagnostics. And we can do all of that fairly quickly. To generate an antibody, target requires a good bit of time.
Yeah.
It's pretty expensive.
It is.
And requires using animals to express something so that you can then get the antibody.
Yep.
That is about a 6-month process. Using aptamers, it's about 2 to 8 weeks.
That's amazing. Wow.
And we're trying to get it down. My goal is to get that down to about 2 days.
Yeah. Yeah. I can see that. I'm sure the technical team is, give me at least a couple of weeks. Yeah. And so, so kind of focusing on this platform approach and, thinking about, from the regulatory aspect, building this body of safety data and then kind of creating your own, design space, using kind of the quality by design terminology from the technical side to say, okay, FDA or whatever regulatory body we're looking at, here is the space in which we have safety data, and then here are all of our targets. Can we go play in the sandbox and really apply this all over the place depending on the needs?
Right.
Yeah, that's amazing approach. And so, David, this has all been great, and I really appreciate, the explaining the RNA origami, the aptamers, the precision medicine that, you guys can bring to the patients and their needs. So let's just talk about the challenges and, advice to other small companies out there. You have a pretty specific, innovative technology. So other companies out there that have, kind of a new technology, how do you get that, moving forward out of the academic, environment and into the regulated?
Right. So I would argue that North Carolina as a whole is actually well situated for that from a startup perspective in many ways. Yeah. And there's a difficulty in North Carolina. We aren't Boston.
Right.
We aren't San Francisco. We're not San Diego.
Right.
It is sometimes a challenge to get funding here. However, North Carolina, more than any other state I've lived in, really has great ecosystem support from the North Carolina Biotechnology Center, which has both grants and loans, to the SBTDC, the Small Business and Technology Development Center, to CED. There's a lot of opportunities for support in North Carolina that's actually really well organized that I don't see in other states. Oh, wow. So from an innovation perspective and moving from a research lab to something commercializable, North Carolina has some significant opportunities.
That's amazing.
And so I would say my recommendation for anybody that's looking is to get involved in those organizations.
Right.
North Carolina certainly has a strong presence with SBIRs, so Small Business Innovation Research Grants.
Yep.
That's definitely something Helixomer has utilized and we will continue to utilize.
Yeah. That's awesome. Well, thank you for joining me today. Thank you for, your eloquent speaking and all the tech talk as well. I truly appreciate the nice balance there. And best of luck to you guys. And we're here to support you throughout the journey, and I appreciate being involved.
Well, so, I appreciate it too. Yeah. I thank you for the time. And we talked about our values, but what we possess, I think Kymanox also possesses, and that's competence, confidence, courage, and conviction.
Oh, that's awesome. That's awesome.
We're happy to be working together. You're allowing us to really expand and move forward where we have been in a holding pattern for a bit.
Awesome. That's great. So thank you for that, and best of luck to you. Look forward to the future.
All right. Sounds good.
That That was Ryan Doxey and David Harrell. For more on Helixomer, head on over to their website at helixomer.com. Thank you so much for listening to or watching this episode. Please subscribe or follow this podcast in whatever app you're using right now, or follow Kymanox on LinkedIn for all updates. This episode was edited and produced by Earfluence. We'll see you again soon on The Factor.